We are witnessing a fundamental shift from simple LLMs to true agentic systems designed to do real auditable work. BioAgent differentiates itself by turning execution into clean deliverables. You don't get a messy chat log, you get a professional folder with readable code, curated plots, and a full execution trace. But this is not about replacing scientists. Agents won't do 100% of the work. We prioritize essential collaboration, giving you the autonomy to choose what to automate while keeping the strategic decisions in your hands. Let's see exactly how this works in practice. Welcome to the BioAgent demo. This is BioAgent. It is not simply another coding assistant or a chatbot. It is a fully autonomous AI researcher that lives right inside your command line environment. Let's start by looking at the terminal. We begin by invoking the agent with a simple init command. Watch how quickly it responds. In mere seconds, it configures its own environment, creates a dedicated workspace, and prepares the underlying infrastructure required for complex biological analysis. Now, let's look at the prompt that I'm typing now. Here's where the paradigm shifts. You don't write a single line of Python. You don't debug pipelines. You simply tell it what you need in plain English, analyze the single cell RNA sequence data in the shared folder. Now, shift your attention to that blue labeled box, Agent's Thoughts. This is crucial. It doesn't blindly execute a script. It thinks, scans the directory, understands the file structure, identifies the raw data, and formulates a step by step scientific plan. Keep watching the terminal text. As it initiates the plan, it realizes it needs the ScanPy ecosystem to handle the specific data set. Notice what happens next. Instead of failing or asking you to fix the environment, which is a common pain point, it autonomously manages its own dependencies, identifies the missing toolkits, asks for permission, and installs exactly what it needs to get the job done. It ensures the system is ready without you lifting a finger. Now, post this, we enter phase 1, that is data preparation. This is the foundation of any rigorous analysis. Let us watch closely as it writes the code. The bioagent is not just loading the file, it is assessing the quality of the sequencing. Bioagent calculates total UMI counts per cell to check sequencing depth. It calculates the number of genes detected to ensure library complexity. Also, it measures mitochondrial percentage to identify dying cells. Let us look at the right side of the screen as the plots would appear. The bioagent generates these standard visualizations of histogram and scatter plots, and it uses visual intelligence to actually look at them to understand distribution of your data. Now, let us focus on the terminal text. It interprets those visual patterns just like a human scientist would. The bioagent determines that the cells are generally healthy but logically decides to apply specific filtering thresholds to remove low quality outliers, making data driven decisions on the fly. Let us focus on the scatter plot that would appear after the filtering now. With the data cleaned, it proceeds to feature selection. Let us now look at these red dots. Those are the highly variable genes it has identified which drive the biological diversity of the specific sample. Watch the transition. It uses those genes to reduce dimensionality, running PCA and computing the neighborhood graph to generate the UMAP projection that you see here. Now, you can freeze your attention on the grid that would appear. This is a critical moment of autonomy. A human analyst usually guesses a clustering resolution. The bioagent tests them. Look at the panels. It runs the Leiden algorithm at multiple resolutions, simultaneously evaluating them to self select the one that offers the most distinct biologically relevant structure. Now, once it selects the optimal structure, it cross references marker genes against its internal knowledge base to automatically assign biological labels. Let's have a look at the dot plots. First step of biological validation. Looking at the grid, on the bottom we have specific marker genes and on the left the cell types. Noticing the circles, the size tells the fraction of the cells in that cluster expressing the gene, while the color intensity shows the mean expression level. This confirms the specificity of the annotation. The bioagent takes the marker genes and maps their expression back onto the UMAP coordinates. The gene expression physically located on the map. It confirms that the marker is not just present in the list, it is spatially restricted exactly to where the cell cluster lives. The violet plot reveals the distribution. Looking at the width of the shape, it shows you the probability density that the gene expressed in every single of the cell cluster and not just subpopulation. Comparing this to the heat map gives you the global architectural view. Look at those distinct blocks of color. Each column is a single cell. You want to see sharp, clear bands of expression that are exclusive to specific groups. See how clean those separations are? That's the final visual proof that the agent's clustering and annotation are not just statistical artifacts, they represent real distinct biological signatures. With the biology validated, the agent concludes the active processing. Look at the terminal summary that would appear now. It provides an immediate executive brief of the run. Read the key results section. It explicitly lists the 8 distinct immune cell types it identified, the native T cells, monocytes, B cells, along with their proportions. And look closely at the pipeline completed checklist. It confirms that every single stage, from data inspection to ligand clustering, was successfully executed. It then finalizes the session. It logs the performance metrics and confirms that the session ID and workspace state have been securely saved. This ensures that the entire analysis is audit ready and retrievable. Finally, let's look at the actual deliverable. The agent generates a comprehensive Markdown report. Let's follow along as we scroll. It begins with the dataset overview to establish the baseline. It moves immediately into quality control, showing you visual evidence for its filtering decisions. It details the analysis workflow step by step, explaining not what just it did, but why it chose those specific parameters. And look closely at the formatting. It weaves technical implementation directly with the scientific findings. You see the code snippets, the generated plots, and the biological interpretation all in one place. This creates a completely transparent, auditable record. It ensures that every insight is traceable back to the exact logic that produced it, removing the black box nature of AI analysis. It takes you from raw unstructured data to actionable biological insight completely autonomously. This is the power of BioAgent.